This is lecture 21 on the basal ganglia. So the brain is always making predictions about what's important in the environment. And so the reason I put this meme up here is that, when you see someone expressing an exclamation and there's something obvious like an airplane, but then the byline is, look at those gas prices. It's something that you didn't expect. And that's the basis of a lot of humor. So it's something that makes sense and is logical, but it wasn't the first thing that you were thinking about. And the basal ganglia plays an important role in this expectation. So we talked last time about the descending motor systems, and we talked about how the cerebellum feeds in and does this sculpting sort of process to fine-tune the movements. Well, it turns out that the basal ganglia provides input to the system too, and it forms another part of a loop, and its job is to initiate and withhold movements, and this is important for sequencing movements. But as evolution has gone on, it looks like it's also then taken an important role in decisions, because deciding to act and when to generate an action or to withhold an action is itself a decision. And so over time, the basal ganglia's role has expanded to making decisions about value and more abstract things. So there are simple decisions, for instance, eye movements. People typically aren't aware of the eye movements, but you're making saccades, that's rapid eye movements around your environment all the time. So here's one image, and it's fairly easy to track people's eye movements. You just put some cameras in front of their eyes and calibrate it, and what you can do is you can produce what's colloquially referred to as a heat map. So where it's red, that means that there's a lot of fixation time, and where it's blue and black, that's a low amount of time. And I've broken this down because this is broken down by the gender of the viewer. So we have men and women, and see if you can guess which is which. So the reason I'm bothering showing you this is that, probably a lot of you think that this is women and this is men, but it's actually the other way around. So it's it's men that are checking out the competition, and the women are more interested in the face and the shoulders. Okay, so why should we care about the basal ganglia? Well, they're a huge structure. This is in rat, and this is stained for acetylcholinesterase, which breaks down acetylcholine. Acetylcholine isn't one of the main neurotransmitters, but it's in the striatum, as are a lot of other things. But this gives you a good indication of how large the structure is. So this is a coronal section. This entire thing is the basal ganglia, or this is the striatum, which is the input part of the basal ganglia. Here's a sagittal section just to show you the approximate size. So this is the thalamus. Here's all of the neocortex. Here's part of the hippocampus. So it's pretty big, right? It's about half or a third of the size of the neocortex, about the same size as the thalamus. It's a major subcortical structure in terms of size, but also in terms of function. You can't live without it. So extensive damage to the basal ganglia and to the striatum. You can't live. Animals can't move. They don't eat. So you can't live without it. Diseases that affect the basal ganglia include Parkinson's disease, Huntington's, even some more neuropsychiatric diseases like attention deficit hyperactivity disorder, Tourette syndrome, obsessive compulsive disorder, and addictions. Part of the reason why is that there's such a rich variety of neurotransmitter receptors in the stratum. These include acetylcholine, dopamine, 5-hydroxytryptamine, aka serotonin, cannabinoid receptors, adenosine receptors, which is the site of action of caffeine, norepinephrine, which is the site of action of amphetamine, as well as the dopamine receptors, receptors for opiates, and even other things like nitrous oxide, histamine, and other things. And the evolution of these structures has been very well conserved across species, and that suggests that it's important to function and it does something very useful for the animal. Again, here's a little Venn diagram that I took off of Wikipedia that shows a variety of psychoactive drugs and receptors. Well, this is the drug table, and the vast majority of these have sites of action in the basal ganglia. Elsewhere too, but basal ganglia often are a central site of action, and so are involved in addiction, both chemical and behavioral addictions. So, what are some of the important functions? Well, it activates appropriate movements. Equally important is that it suppresses inappropriate movements, and it's very critical for sequential action. So, if you need to do something in a certain order, the basal ganglia is important for withholding a particular part of the action until it's called for in the sequence. So, that's in the motor control. Um, but it's also important for making choices, as we'll talk about a little bit later, particularly by learning from past rewards to guide future actions. And this is a form of what's known as reinforcement learning. Now, I don't have the animation isn't quite working here, but what this, let's see if it works. No, it can't play. So what normally I do here is I play this animation, and the circle lights up at a regular time, and so it's a rhythm, and I have people clap along, something like that. And within a few cycles, people can anticipate when the light comes on and clap right as the light comes on. This other one, I have the timing in between the illuminations is unpredictable because it's random, and therefore you're in this pure feedback loop. And so if we make a histogram of the times at which people clap, when it's periodic, people can anticipate it, and the distribution of clap times falls right around when the light comes on. But in this case, you have to wait for the thing to come on before you clap. And so it takes time to process the visual information, generate the motor command, and actually clap. And so there's a delay of 100 milliseconds or something like that. So this is what I mean by, or this is a simple case of predicting the future. And the way you do that is you build a mental model of the world. And again, this is a fairly simple one, but we have very complicated models, and we use these all the time. We talked last time about the control of movement and how having this efference copy that's used as part of a forward model is very important, and it involves hundreds of muscles and joint angles and vestibular input and the proprioceptive input. So there's a lot of information that's coming in, and that's what allows humans and other animals to have very fine motor control. But being able to anticipate other things, like when am I likely to be attacked, has tremendous survival advantage. So we talked about the motor domain, but also in the cognitive and limbic domains. Hey, if I eat too much of that, I'm going to feel sick. So even though it's delicious, I should stop eating. So that would be, in this cognitive and limbic domains. More specifically, the basal ganglia are very important for learning associations between environmental states, actions, and their outcomes, their affective outcomes. Did it make me feel good or did it make me feel bad? The basal ganglia are important for assessing if thoughts and actions have high value or have low value based on past experience. And by doing that, the basal ganglia help select the good actions and suppress the bad actions and thoughts. And so this is why when we see pathology of the basal ganglia, not only do we get impairments in motor activity or motor control, so think like Huntington's disease or Parkinson's disease, but we also see problems in thoughts. So you can think of things like obsessive compulsive disorder, also negative rumination that occurs in depression and anxiety. So this just summarizes a little bit. Well, here's a guy who's going to go taunt a crocodile, and you might think, well, that's not a great idea because this might happen. So if you can anticipate that, hey, there might be a very negative consequence to my action, I should withhold those actions. Again, that confers a benefit for survival. But you can also have too much of a good thing. So if there's cookies and they're delicious, and it's ahead of the your family's party, and you go and eat the whole tray, it might taste good while you're doing it, but in the future your stomach's going to hurt, your family's going to be mad because you ate all of the cookies. So being able to anticipate what's the consequence is important for making good decisions. Okay, so let's talk about the basal ganglia. The input is called the stratum, and in primates it's broken up into 3 different structures or names called the caudate, the putamen, and the nucleus accumbens, which isn't in this section. It's in a more anterior section. So that's the input part. This purple part is the globus pallidus. We'll see this on one of the next slides. You'll see it in better detail. But the basal ganglia also comprise the substantia nigra, which is shown here, and this is in a separate section because it's more posterior than these sections. Importantly, what we'll see is that the basal ganglia gets input from the neocortex from nearly the entire cerebrum. So everything that's in purple sends projections to the striatum, which again is the input of the basal ganglia. And this is everything except for the primary auditory cortex and the primary visual cortex. But as soon as it goes to these belt areas or extrastriate areas, then all that information goes to the basal ganglia. So the basal ganglia essentially has a survey of the entire neocortical state. That's about a third of its input. The other third comes from a mixture of the thalamus, the amygdala, and the hippocampus. So it's integrating all this information about the state of the brain as signaled by the neocortex, by the amygdala, which has to do with fear, desire, and with the hippocampus that has to do with spatial location and memory. So it's integrating all these things together. So here is a simplified connectivity. I don't expect you to know all the connectivity. What I do want you to know are the two main pathways. And these are referred to as the direct pathway and the indirect pathway. So the direct pathway, we have inputs coming from the cortex into the putamen, the caudate, or the nucleus accumbens, collectively referred to as the striatum. So it's coming into here, and the direct pathway, like this neuron, goes right to this what's called the internal segment of the globus pallidus. Here's the globus pallidus. It has an external segment and an internal segment. So it goes from the striatum to the internal segment, and then it goes to the thalamus. That part's not being shown here. This is the direct circuit. And what this does is this initiates movements. So you can think of this as the go circuit. There's another circuit, which is the indirect circuit, and this has 3 inhibitory synapses. So we have the cortex, the cortical input coming into the striatum. That's excitatory. Then we have from the striatum to the external part of the globus pallidus, the external part to the internal part, and then to the thalamus. This is an inhibitory one, this is inhibitory, and that's inhibitory. So in the direct pathway, we have 1, 2 we have this from the stratum to the internal part and the internal to the thalamus. Those are both negative. So 2 negatives make a positive, and that's why it's the go circuit. Whereas the indirect pathway has 1 negative, 2 negatives, and then out to the stratum or to the thalamus is the third negative. And so that has a net effect to suppress movements or to stop movements. So that's what I want you to know, the direct pathway and indirect pathway, and that the direct pathway initiates movements, indirect stops them. And we'll see how that works here in a minute. You don't have to recapitulate the circuit or anything, just know the direct and indirect pathways. So within the basal ganglia, there's a huge amount of convergence. What does that mean? Well, the cortical neurons innervate, provide about 0 to 40 synapses onto a spiny neuron, and one spiny neuron gets input from about 11,000 cortical neurons. And the reason they're called spiny neurons, well, they're medium spiny neurons. That's the principal cell of the stratum. The soma is a medium size, and it has these dendrites that are covered with spines. The reason it's covered with dense spines is because it's receiving so many inputs. So again, it's getting about 11,000 inputs. There are about 3 million striatal neurons in the rat. The GPE has about 46,000, and you can see that the numbers of neurons fall off quite drastically. And so by the time we're looking at the output structures, the SNR, SNC, and GPI is not in here, but you're talking about 30,000 neurons. So you're going from 30 million down to about 30,000. So it's a high, many orders of magnitude convergence. So what the basal ganglia are doing is that they're integrating information from across the cortex, amygdala, hippocampus, thalamus, and it's sending it out through this about 30,000 plus a few more neurons. Okay, so we want to. I'll show you how the direct and indirect pathways work. There's the cursor. Okay, and so they form loops. So we start with a cortex that goes to the striatum. The direct pathway goes right to this GPI, this internal segment, which goes to the thalamus, which then goes back to the cortex, forms this loop. The indirect pathway, we have cortex to striatum to the external segment of the GP, then to the internal segment, then to the thalamus and back. Sometimes instead of going to the GPI, it goes to the reticular part of the substantia nigra. It's not a detail you need to worry about. Okay. So let's see, how does this work? So let's look at the direct pathway. So when the cortex is active, if it were to activate a medium spiny neuron in the striatum, it becomes active. It inhibits a cell in the globus pallidus in the internal segment. So you see this dip. So spiny neuron fires, GPI neuron gets inhibited. The GPI neuron is inhibitory onto the thalamus, and it's normally active. So even when it has no input coming in, it's normally active. So when the spiny neuron activates, it causes it to pause. That releases inhibition from the thalamus, so the thalamus can then burst its activity, sends that up to the cortex, and causes a burst of activity. For instance, if this is in the primary motor cortex, then that would initiate the movement. Well, let's look in the indirect pathway. So if we activate a spiny neuron in the indirect pathway, because of this double inhibitory pathway, it inhibits a cell that's inhibiting this one. So it's a double inhibitory pathway. We actually get an increase of this GPI cell that increases the amount of inhibition on the thalamus, causing the low level of background firing to suppress. That suppresses the cortex. And so that makes it even less likely that the movement is going to be initiated. Or if there's a movement ongoing, this stops the movement. So that's how the direct pathway can promote movements. The indirect pathway can inhibit movements. Okay, so can we see any evidence of this? Well, if we go in and inject a GABA agonist into the region of the substantia nigra, and again, that's the part it's kind of equivalent to the GPI, the internal segment of the globus pallidus. So if you inhibit that, you're inhibiting both the direct and indirect pathways, but it has more of an effect on the indirect pathway, and you start to see these spontaneous saccades to the left visual field. Right, so the eye movements start to become less controlled. In this task, the animals are supposed to fixate right on this center target. So they shouldn't be doing this, but they do it anyway. So these are in well-trained monkeys. So if that's the case, we would expect diseases that affect basal ganglia to produce motor deficits, and that's what we see in Parkinson's, Huntington's, and some other pathologies of the basal ganglia. For Parkinson's, the first line of treatment is L-DOPA, which increases dopamine levels. So Parkinson's disease is unusual in that we know exactly what's happening. It's the progressive death of dopamine neurons. We have some ideas of why they're dying, but we can't really stop it or reverse it. So because dopamine neurons are dying, what we can do is try to increase the level of dopamine that's being released by the cells that are still alive. So what we do is you can give L-DOPA, which crosses the blood-brain barrier, gets taken up, and this is like putting more precursor into a chemical reaction. So the L-DOPA is the precursor that gets converted to dopamine, and then the neurons, the dopamine neurons that are still there, release more dopamine. The second line of treatment is to actually stimulate the basal ganglia or the thalamus via deep brain stimulation. And you can look up some videos online if you want. This is a deep brain stimulator. The battery pack and controllers, are down here in the chest. There's leads that come up, they go under the skin, and there are some electrodes that are implanted. Oftentimes it's in the globus pallidus, or sometimes it's in one of these other structures. It's called the subthalamic nucleus that I didn't talk about. And what happens is that when you turn this on, it disrupts a pattern of rhythmic activity that spontaneously forms in the basal ganglia. And this rhythmic activity provides or promotes tremor. And so this is a video of a guy that has a little, he has a remote control. He stands up and when he turns off his deep brain stimulator, you see this tremor. And when he turns it on, it goes away. So if you're interested in seeing that, you can find a video online. So what does this look like in terms of neural activity? Well, we can't really record neural activity in a human except under very rare circumstances, like when there's a patient who's getting an ablation for some purpose like epilepsy. There'll be a recording electrode that gets put in, and it's just there for a dozen minutes or something to map out the area. So instead, we often have to rely on animal models, and this is from a monkey. And this is a monkey that has received a drug that has killed off some of the dopamine neurons. And so it's a model of Parkinson's disease. And what you can see is that you start to get this rhythmic activity. And this rhythmic activity in the neurons is what promotes the motor tremor. When you turn on the stimulation, you see that it breaks up that rhythmic activity. And when you turn the stimulator back off, you're back to this rhythmic activity again. Interesting story, this compound, which is called MPTP, is a toxic byproduct of opioid synthesis. And this was discovered because a chemist in California, who I think was a graduate student, tried to synthesize heroin and did so, but also had some MPTP in it. And so within a few days after taking the drug, essentially became Parkinsonian, went to the hospital. And of course, it's highly unusual. And so it was eventually discovered what compound had done that. Okay, so what's happening in Parkinson's disease? I don't expect you to know all these different pathways, but to a first order approximation, the D1 receptors increase the striatal neural activity in the indirect pathway. The D2 receptor activation inhibits striatal neuron activity in the indirect pathway. And so when you start to lose dopamine, essentially what happens is that you're reducing the ability to turn on the direct pathway to initiate movements, but you're also taking away the ability to inhibit the inhibition via the indirect pathway. And so what comes out is this sort of unfortunate paradoxical situation in which subjects have a hard time initiating the movements they want, but they also can't inhibit movements that they don't want, like this tremor. And you can read about this in the book, but you can see that the indirect pathway sort of becomes potentiated and gives you this dyskinesia. Okay, I'm not going to test you on Parkinson's disease. But what is important to know is that, so Parkinson's disease, it's not just a motor dysfunction. Let me clarify. I'm not going to test you on the nuances of those pathways that's on all this stuff. I just want you to know the direct and indirect pathway. Okay, and I want you to know the dopamine stuff that we're going to talk about next. But Parkinson's disease, so it's not just motor dysfunction, you also see cognitive deficits related to it. So these are mood disorders and also problems in attention and motivation. For whatever reason, the dopamine neurons that are projecting to the motor parts of the striatum are affected first, or are affected to a greater extent, so that the disease is typically diagnosed when tremors and other sorts of motor problems occur. And when the disease is in its early phases, you can give someone L-DOPA. That helps alleviate the tremors and allows people to activate the movements what they want. But oftentimes it also has effects on motivations and decisions. And this is oftentimes sexually dimorphic. So older men on L-DOPA and other types of dopaminergic drugs tend to become hypersexual. So you can think of the sort of stereotypical lecherous old man that's harassing the nurses. Women, on the other hand, instead of becoming hypersexual, oftentimes become compulsive gamblers. So they'll play bridge or bingo or those sorts of things. And why that has the sexual dimorphic property, nobody's exactly sure. But why does that happen? Well, you're losing dopamine here in the motor parts of the striatum, so you increase dopamine. Dopamine was typically at normal levels in the limbic part, so this is the part mediating emotions and desires for things. Now you're boosting it. So it's kind of like applying cocaine or another type of stimulant to this circuit, the limbic circuit, because now the limbic circuit is in a hyperdopaminergic state where the motor part is in just a normal dopaminergic state by giving L-DOPA. Okay, and this refines this idea a little bit more that we talked about these loops that go from the cortex to the striatum to the GPE, GPI, and now to the thalamus, and then these go back to the cortex. And so some of these loops we talked about go through the motor part of the cortex, some go through the sensory parts of the cortex, some the associative, and some limbic. So this is a motif that you see in different parts of the brain. So it's not just in the motor domain. And this is why you see, motor effects, sensory effects, and limbic effects when you have drugs that affect, or diseases that affect how the basal ganglia are working. This is a famous paper mapping this out in rats. And so you can see that sort of this ventral medial prefrontal cortex is going to this ventral part of the stratum. This is the accumbens, so this whole part that we didn't see in the primate because it was in a different section. Here's the sensory motor part, which is out here on this more lateral part. And the cognitive part is sort of this bluish purple in the middle. And again, this is why if you disrupt this part of the circuit, you get akinesia, tremor, you get motor symptoms. If you affect this part, you start to get disruptions in associative and sensory processing. So think like attention deficits or compulsive thoughts. And if it's in this limbic part, this is associated with either apathy or addiction. So having to do with motivation, either too little motivation to do anything or too much motivation for certain things like drugs of abuse or other types of behavior. And so we're showing these sort of operating in parallel, but the recent evidence suggests it's more like a spiral, so that you have sensory information coming in that goes to the associative loop, goes to the limbic loop. So you have information coming in, you're making associations with that information, you're sending it through the limbic loop to decide if that's something that's highly motivating for you. And if it is, then you activate the motor circuitry. So you have this sort of spiral or hierarchy that goes from getting input from the periphery, getting associations, forming an assessment of how much you want that thing, what impact it's going to have on your affective state, and then do something about it. So control the motor action. So let's see how this works as an action selector. So we're not going to talk so much about the motor domain. We did that last time. But I'd like to illustrate now how this system can use past experience with rewards and punishments to activate or suppress certain movements, which embodies a choice. And this is kind of a silly binary example, but it seems to hold true. And I think this is more clear than some other examples. So imagine that you go to a restaurant and you're faced with ordering either the steak or the fish. So what we know about the system is that the neocortex has a bunch of cells, and it's encoding different features like the price and what does it look like and the description. And so these are sending projections down to these medium spiny neurons of the striatum, and they have different sized synapses. So some are strong, and I'm showing those with a big circle. Some are weak. I'm showing those with a small circle. And something we didn't talk about yet is that these medium spiny neurons, so they're GABAergic, and they send collaterals to one another. So what that means is that if this one's active, it inhibits this guy, and if this one's active, it inhibits that guy. And what this does is it helps have only one active. So you don't want to pick both the steak and the fish. And so this mutual inhibition is a way to just get a singular output, to make an either or decision, not both. And what we know about dopamine is that it's important for modulating these synapses, right? So we talked about associative learning, but now we're faced with this problem that you make the choice in the restaurant, and then, if you're lucky, it's several minutes and not hours. Later, you get the food and then you get to taste it. And this is what's known as the temporal credit assignment problem. So things you did in the past affect your state now. And dopamine seems to be a signal that helps to solve this problem. And what dopamine does, we talked about this before, it does this reward prediction error signal. So if all of a sudden you get an unexpected reward, dopamine neurons fire. Right, so so dopamine neurons are signaling this reward prediction error signal. It's the difference between what you got from what you actually expected. Here you didn't expect anything and you got a reward, and so you get this uptick in reward. Sorry, my cursor's not behaving well. Right? So you didn't expect anything, you got something. So something minus your nothing expectation is a positive. Here, a cue came, so you expected a reward. So you got exactly what you expected, so dopamine neurons don't activate. But if you weren't expecting the conditioned stimulus to come on, like a beep, then dopamine neurons fire to the conditioned stimulus. Now, if you give the conditioned stimulus, but then don't give the reward, you expected something but got nothing. So 0 minus something is now a negative. Okay, so this is the reward prediction error signal. And what we'll see is that, well, let's walk through this. So imagine that when you went into the restaurant, that the medium spiny neuron encoding for fish, and again, it's not just one cell, but it would be a population of cells, but we're just going to consider this as one cell. So imagine that all the cells, or the one cell that represents the fish population, had more inputs coming in. It had more excitatory input. This is all glutamatergic. So it activates, it suppresses this guy, the steak, and so fish is the winner, meaning that the basal ganglia sends back to the cortex, hey, you should pick the fish. Okay, and then imagine once you get it, and it tastes like an old rubber boot fished out of the river. It tastes horrible. Well, you expected it to be good. It tasted terrible. So you have a negative reward prediction error signal. And what this does, when this dips, this actually causes LTD, long-term depression of these synapses. So now notice how I made them all smaller. So now when you're back in the restaurant, the medium spiny neurons encoding for steak have more input, more excitatory input than do those for fish. So now you pick steak the next time. So you've learned by experience. Okay, but let's rewind. So imagine, the first time you've been in the restaurant, you get the fish is the winner, and it's amazing. It's some of the best fish you've ever had. Well, you still get a reward prediction error signal, but this time it's positive because you expected kind of mediocre fish, but it was great. So it violated your expectation on the positive side. And these bursts of dopamine actually facilitate long-term potentiation. So now we're going to make these bigger. So now when you're back in the restaurant, it's much more likely that the striatum will say, yeah, get the fish, because in the past that gave us a dopamine spike and did LTP. So now this guy has an even bigger input than the steak neurons do. Now, of course, it's a lot more complicated than this. So this would be if you didn't have any other thoughts and it was just a pure sensory to choice process. But instead, you have all these, the cortex is doing these other things like, should I try something new? I don't like fish. I'm on a diet. It's too expensive. The steak is too expensive, or the fish is too expensive, whatever. Or it has some ingredient I don't want to eat. You have memory. The hippocampus might say, oh, well, we had steak yesterday. So all these things actually go into your decision as well. But you can use the same process to represent all of these factors. And so this is known as reinforcement learning. And this has triggered an entire branch of artificial intelligence, also called reinforcement learning. Because even though it's a relatively simple algorithm, you're taking the difference between what did you expect and what did you get from what did you expect, but it can solve all sorts of problems, from doing chess to picking foods, to picking stocks, to even doing very difficult motor control problems like balancing. This is called a double pendulum. So there's a pivot here and a pivot here. And so it's, humans can't do this, but using reinforcement learning, you can control this robot to actually balance these things. And there are videos of that if you want to look those up. And it's, a multi-billion dollar business now. Okay, now I want to talk about reward. So, rewinding just a little bit, right? What this means is that if we're potentiating inputs that have been associated with better than expected rewards in the past, that you might expect these cells to be encoding something related to reward. And we actually do see that, and it can change fairly rapidly. So now what this is data that was recorded from a monkey that's performing a saccade task, meaning that he stares at a central fixation point and then just has to move his eyes to an illuminated target. And this is showing the response of medium spiny neuron to the illumination of a target. And the animal knows ahead of time that, so there's up, down, left, and right, and this is to the up, we'll say. In this block of trials, when it's indicated with that, the animal knows that when he makes a saccade upwards, he gets a drop of juice, so he gets reward. In this block of trials, when he looks up, he doesn't get a reward, but he has to look up to get to the next trials, say the one to the left, in which he does get rewarded. So this is the response of the same neuron to the same stimuli, but in one case it means you're going to get a reward, in the other case it means you're not going to get a reward now, but you have to do it to get a reward in the future. And what you can see is that on the rewarded trial, it's higher amplitude and longer duration. And so this probably has something to do with both learning, but also the fact that dopamine neurons are more likely to fire when the light comes on and it indicates that a reward is upcoming. So if the animal can't expect it because it's randomized the targets between the up, down, left, and right conditions. So we can't unwind, is this the direct result of dopamine modulation, or is it because of long-term potentiation when the trials occur? So it's probably both. And we can see this also in humans. So if we do functional magnetic resonance imaging and look for blood flow signal, the bold signal, we can put subjects in the scanner and then ask them this question. Would you like $20 now, or would you like more than $20 sometime in the future? Or you can ask the question, do you want $20 now or $20 in the future? And here's how much they are likely to value one thing over the other. And the way you do this is you say, okay, here's a coupon for $20. When you bring it back to us, we'll give you $20. In this case, as soon as you're out of the scanner, you hand us the coupon and we'll hand you $20. In this case, right, the 60 days, that means you have to come back in 2 months, give us the coupon, and we'll give you $20. And out here, it's a half a year. And so then what they'll do is they'll say after the experiment, well, if you can't redeem this until 2 months, how much are you willing to sell it for? And so this is, about 0.6. So 6 out of $10. So they would be willing to sell it for $12 now. And if it's a year out, then they'd be willing to sell it for 40%. So that would be $8, right? So the farther something is into the future, the less value it is now. Okay, and so what you can do is you can get these, what it's called a delayed discounting curve, and then you can ask where in the brain do the signals follow this. So you're giving them coupons and telling them when the redemption date is, and then you can look in different areas and find, is there a signal that correlates with this? And where you find this is in the cingulate cortex. You find it in the, you find it in the ventral part of the striatum, and you find a little bit in this posterior cortex. And these things are all hooked up. So the anterior cingulate projects into the striatum, and this part of the posterior, that's posterior cingulate, sorry, not the posterior parietal. So we have the cingulate cortex and we have parts of the striatum. We can also ask, okay, not only delay discounting, but what about probability discounting? So now you don't have a coupon, but you have a lottery ticket. So the lottery ticket has both odds and when you can redeem it. So some might be a lottery ticket with 80% odds and it pays out today. That would have a high value, or you could have one that has a high probability, so 80%, but you can't redeem it for a year. So it's discounted by delay. But you can also have a coupon or a lottery ticket that is redeemable today, but it only has a 0.1% chance of paying out. And again, that's now discounted by the probability. And so if you ask where in the brain cares about this conjunction of delay and probability, the biggest place that lights up is the ventral striatum. There's a little tiny part of the orbitofrontal cortex as well. So the bottom line is that the striatum cares about valuation. Now, what about drug addiction? So this ventral striatum is a key place for mediating the wanting of something, not necessarily the liking. So we talked about this before when we talked about dopamine. And now today I'm telling you about how dopamine can implement this reinforcement learning. So there are drugs that elevate dopamine very high, much more than the normal release would. So what would happen there? So here what we're looking at is dopamine release, and what we're looking at is 2 cues that a drug is about to come up. right? In this case, it is future cocaine. So this is cocaine. So they're going to turn on a light, and then they're going to give them cocaine like 10 seconds later. So like at the end of this little green line here, okay? In a naive animal, they don't know this association, and so dopamine doesn't go up. In the trained animal, they know, oh, here's the cue that says I'm about to get cocaine, so dopamine starts pouring out, okay? Now let's look at the striatal neurons, and this is for just a natural reward. So here's water. So this is a thirsty animal. Some of the neurons do this, so they ramp up. This is when they're getting the water. Some are low, and then they give this little spike after the water. Some pause when the water comes. The reason I'm showing you this is that when we look at neurons that fire to cues for cocaine, they look very similar to those responses for natural rewards like water, right? So this one kind of looks like that one. Here's one that kind of ramps up like that. Here's one that kind of pauses like that. So drugs of abuse seem to tap right into the system that encodes natural rewards like food. So not only do drugs of abuse, cues for drugs of abuse cause the release of dopamine, like we know they do for food, but the striatal neurons themselves are encoding the cocaine-related cues and cocaine themselves similar as they would for food. So it looks like drugs of abuse are essentially hijacking the systems that are there for natural rewards and essentially cause learning in the system. But because the dopamine goes so high, it's like, hey, you got a much better reward than you were expecting. And so let's increase the synaptic plasticity. Let's undergo LTP for those neurons that were involved in processing information about the drug. So the cues and the actual taking. And the result is that you get fixation on drug-related cues and you fall into habitual drug-taking activities, and it becomes very, very hard to unlearn those things. So essentially you'd have to go in and somehow depress, undo the long-term potentiation of just those selective neurons. And that's really hard to do. We have drugs that can induce LTD, but that does it across all things, not just the cells that are involved in the drug-related cues and drug taking. Okay, and that does it for today, and here are the important concepts.